Okay, let's get started because we've got lots to do. Okay, just a couple of reminders as we get started today. Chapter 10 homework is due, so if you didn't put it in the box yet, make sure you do it before you leave. The quiz is open on Moodle until 11:59 p.m. tonight, so make sure that if you haven't done it already, that you are writing down to do that. And as a reminder, next Wednesday is our test, so we'll be covering chapters 9 and 10. So today we're going to be working on photosynthesis again, and we're going to be finishing that up on Monday. So that's where we stand. Any questions on that? Okay, so here are some questions to think about from last time. Take a minute to talk about it with your neighbor or talk about it with yourself. What are the reactions for photosynthesis? How are mitochondria and chloroplasts similar? And what color light should you shine on your plants and why? So take a minute and talk about that. Get us back into photosynthesis mode. Okay, what are the reactants for photosynthesis? If we're looking at the basic equation, what are the reactants? Carbon dioxide. Oh, okay, sorry, I can't do that. I just learned, oops, sorry, I didn't learn. I just learned I can use the end of this as an eraser. I'm getting there. By the end of the semester, I will have, yes, thank you. Okay, so carbon dioxide, somebody else said energy. Oh my gosh, okay, I didn't learn about not putting my hand on your stuff. What is the form of energy that we're using? I heard two answers. What is the form of energy that we're using? For photosynthesis. We're gonna use like ATP is involved and we're gonna use it too, but the original source of energy is light. And what's the last reactant? Water. Okay, how are mitochondria and chloroplasts similar? Multiple membranes, so they both have that outer and inner membrane. What makes the chloroplast different from the mitochondria in terms of membranes? The chloroplast has what else? It has an outer membrane, it has an inner membrane, and it has that thylakoid membrane. Remember those stacks, the grana, the stacks of thylakoids, the pancakes or the cookies, whatever you want them to be. Anything else that's similar? We haven't talked about this. They're both going to involve some kind of electron movement, some kind of electron transport chain, which is what we're going to get into details about today. Okay. And they're both involved with conversion of energy from one form to another. What color light should you shine on your plants? Red and blue, or red plus blue makes purple. Why? Because it's the best color? No. Because chlorophyll is going to absorb red and blue light best, and it's going to do what to the green light in the middle? If it's not absorbing it, it's going to reflect it. Okay, so we're shining purple lights on our plants. And that's kind of where we left off on Wednesday. We were talking about what happens when light hits chlorophyll. So chlorophyll being able to absorb that light. When light hits chlorophyll, it excites those electrons. And so it's going to move it to a higher state. And then we looked at this idea of an isolated chlorophyll molecule. So if we take chlorophyll out of the chloroplast, it'll actually do this. And there's a picture in your textbook that shows you that when it's excited, it goes to from the ground state to the excited state. It's unstable and it's going to fall back down, and it's going to release that energy that it had in the form of heat and in the form of light. So if you look in your textbook, there's a picture of a beaker of chlorophyll that's bright red, because that's showing what would happen when those excited molecules become unexcited, when they're not excited anymore. And I think this is where we I don't think we talked about this really at all, right? This is where we left off. So this is showing you what happens with light when light hits chlorophyll in an actual chloroplast in the thylakoid membrane. So we're in the thylakoid membrane, same structure as all the other membranes we've been talking about. We've got this embedded protein structure here, and this is what we call a photosystem. And this is just composed of two different parts. So we've got on the outside, this big purple molecule on the outside, which is not really purple in a cell, are the light harvesting complexes. And then in the center, this lighter purple molecule, periwinkle, whatever color you want to call it, is the reaction center. Complex, I guess I should say. So on the outside, what do you think these little green blobs are? Those are pigment molecules. So those could be chlorophyll A, could be chlorophyll B. These are the pigment molecules. So all these little dots are pigment molecules. And you can see over here, we've got this photon of light coming in. It's not writing anymore. So we've got a photon of light coming in, and it's going to hit a pigment molecule. So it hits this first pigment molecule, and what's it going to do? It's going to transfer energy, just like we see here. It's going to take these ground state electrons and move them up to the excited state in the pigment molecule. So it's going to make them bounce around. So this is my electron. And it's going to get hit by some light, and it's going to go to a higher energy state. This is a really cool ball that might hit the ceiling if I throw it hard enough, but I'm not going to try to do that. So this energy is going to come in, it's going to excite that electron. That electron is going to transfer some energy, and it's going to bounce around, literally bounce around from one pigment molecule to another pigment molecule to another pigment molecule to another pigment molecule until it hits these special pigment molecules in the middle. So this is a special pair of chlorophyll molecules. We're talking about them generically in a second. We'll talk about photosystem 2 and photosystem 1. It's going to hit those, and these are going to get excited also, and they're actually going to release electrons. So they're going to release electrons to be accepted by this molecule, which is probably not really a periwinkle square in an actual photosystem, but this is called the primary electron acceptor. So the light harvesting complex is harvesting the energy from the light. It's bouncing around until it gets transferred into the reaction center complex, where those chlorophyll molecules will transfer the electrons to a primary electron acceptor. Okay, where is this happening? This is in the thylakoid membrane. Okay. So the different pigments and the number of pigment molecules will determine how much light is able to be harvested, or light energy is able to be harvested. And again, these pigment molecules could be chlorophyll A, chlorophyll B, they could be carotenoids, any kind of pigment, but we're showing them as green, so they're chlorophylls. So we're going to proceed into the light reactions, which we've kind of alluded to, but we're going to get into the details of the light reactions now. So you'll see here, this is the system that is involved in the light reactions. You remember, when we think of the general equation for the light reactions, what's coming into the light reactions? If you think back to this slide, we're on this light. Why is it doing this? We're on this light reaction side. And we have one of the reactants for the equation. Somebody said it. Water. So we have water coming in. So water is our reactant here. And what are we going to produce from this water? We're going to produce some oxygen. That's kind of the photosynthesis equation is really separate, like two different things are happening. So in the first part, in the light reactions, we're taking water, we're splitting that water, we're getting oxygen. Obviously, something else has to be produced too, because we have oxygen here and oxygen here. These guys can't just disappear. So we're going to get some hydrogen ions. If we have hydrogen ions, what else are we going to have? Electrons. And then, so we'll talk about what we're going to do with those electrons in this process. So again, we're talking about these photosystems. These are the two different photosystems. We have photosystem 2 and photosystem 1. They're very similar in structure. You'll notice the difference here is in the pair of chlorophyll molecules. So in photosystem 2, we have chlorophyll molecules that are called P680 molecules. What do you think that 680 is representing? Somebody, I think, might have muttered it. Somebody might have muttered it again.
Wavelength.
Yes, the wavelength. Okay, remember the spectrum of visible light? We're in that spectrum of visible light. P680 is in the red range. So these are molecules, chlorophyll molecules, that are best able to absorb the 680 wavelength. When you did the beat experiment, I think you were reading, no, you were in the 500s, I think, for that one. But on the spectrophotometer, you can read at specific levels to read absorption. That's what you were doing with the beat experiment. That's what you did with the enzyme experiment. You were reading at specific wavelengths. This one would have the highest absorption at 680. So it's absorbing best at 680. Over here, you'll see we have P700. This is a more red, far red. Again, it's still chlorophyll molecules. Their absorption is just best at that 700 wavelength instead. So we're going to go through this step by step, and we're going to keep track on our yellow charts. We are going to deal with only 2 steps. So we're starting with the light reactions. So light reactions is a step. There are multiple steps within the steps, but we're just going to keep track of the big picture on the yellow chart. So we have the light reactions taking place where? Thylakoid membranes. We've talked about some of the things that are going to go into it. So water goes in, light goes in, because we're dealing with the light harvesting complexes of the photosystems. And we'll talk about what we're going to get out of this. So step 1, we're going to start at the beginning. What's coming in? Because it's the light reactions, we know it's light. Okay. Seems more sensitive today. So light is going to excite some electrons. It's gonna move them from the ground states, and they're gonna transfer that energy. It's gonna bounce around between these pigment molecules. So that energy is gonna move between pigment molecules until it gets to the P680 molecules in the reaction center. So it's starting in the light harvesting complex and bouncing around until it gets to the reaction center. When it gets to the reaction center, those P680 chlorophyll molecules are going to transfer an electron to the primary acceptor. So the P680 is going to transfer those electrons to the primary electron acceptor. This is going to make P680 into P680 plus. Why does that make sense? It was electrically neutral. It's losing a negative charge, so now it itself is going to have a positive charge. So that's going to make it available and ready to do what? Take another electron. Okay, so every time it's transferring electron, that makes it available to take a new electron. So when it loses this electron and becomes P680 plus, is it reduced or oxidized? If it's losing an electron, it's oxidized because oxidation is lost. So P680 becomes oxidized, and then it will become reduced again immediately because it will gain an electron again. Okay, so that's step 2. We've transferred the electron to the primary electron acceptor, and that'll happen twice because there's 2 molecules. In step 3, you can see over here. Step 3. Sorry, we're really struggling with this today. Water is going to be split. What's going to make this reaction happen? Because what makes every reaction happen that we talk about? What is always the answer? Enzymes. So there's an enzyme that's going to split water into hydrogen ions, electrons, and oxygen. So we're breaking up the water. These hydrogen ions, we're going to get 2 of them. They're going to go to the thylakoid space. Now I don't even know what I did. Oh, wow, I went to the end. Okay. I need more space. I know that the end erases, but I still haven't mastered this. Okay, so these, we'll just go up here. These hydrogen ions are going to go to the thylakoid space. These electrons are going to go to the P680 to reduce it. And then the oxygen, the half oxygen, the half of the O2 is going to combine with another half O2 to become oxygen, which is then released. So we've split water, we've made some oxygen, and we've given electrons to the P680 because it can accept it because it has donated the electrons. So the splitting of water is providing the electrons that are needed for the P680 to transfer to the electron acceptor. Okay, take a minute to tell someone next to you what happens in photosystem 2. Okay, somewhere in your conversation, you might have thought, or somewhere in your brain, you might have thought, why are we starting in photo system 2? And then going to photo system 1, or maybe you didn't. I always think that. I'm like, what? Why are we starting with 2? That's because this is the order they were discovered in. So we discovered photo system 1 first, so they named it, hey, photo system 1, and then they're like, oh, there's another one. Well, shoot, we already called that one photo system 1, so we gotta call this one photo system 2. So it goes in reverse order. Okay, so at the end of this photo system 2 reaction, we've released the oxygen. We've got some hydrogen ions moving into thiazoid space. What are we going to create with a bunch of hydrogen ions? Just like we've done in cellular respiration, if we get a whole buildup of hydrogen ions, what do we make? We're going to make a concentration gradient where we've got a whole bunch of hydrogen ions here and not so many hydrogen ions here. And that's going to be helpful to us. We also have some electrons, and we've transferred these electrons to the primary acceptor, and we're going to move into step 4. And step 4 should look kind of familiar to you. You see PQ, we've seen Q before, cytochrome we've used before, and at the end, you see that we're producing something that should look familiar, which is, not in the end, it's actually in the middle, but. Okay, so we're going to take these electrons, and step 4 of this light reactions is actually an electron transport chain, just like you would see, similar to what you would see in cellular respiration. So we have electrons, and we're going to move them down through a series of molecules, and we're going to use their movement to pump hydrogen ions to make more of a gradient, and then we're going to use that hydrogen ion gradient to make ATP using what enzyme? ATP synthase, okay? So step 4 is the electron transport chain. We're going to transfer electrons from photosystem 2 to photosystem 1 using a series of carriers. And in this process, we're going to pump hydrogen ions. So we're going to transfer from the primary electron acceptor, transferring electrons to this mobile electron carrier, to the cytochrome complex, to another mobile electron carrier, which is going to eventually get them to photosystem 1. So again, and we'll watch a video in a minute showing through how they move through. As this happens, step 5 is going to happen, which is our ATP synthase. We're going to generate some ATP. And again, this is through the process of chemiosmosis, which again should be familiar to you because that's the terminology of building up that hydrogen gradient, the hydrogen ion gradient. And ATP synthase is the enzyme that's involved. And again, we'll talk about that more on the next slide. So we're moving a bunch of electrons. At the end of the electron transport chain in cellular respiration, who takes the electrons? What is the final electron acceptor in the electron transport chain? It's oxygen, but that's not the case here. So there are differences in this electron transport chain. We're going to get a similar outcome in that we're going to get ATP out of it. But these electrons, instead of being transferred to oxygen at the end, are going to be transferred to that P700. So the electrons are going to be transferred there. And simultaneously, we still see some more light coming in. In step 6, we see some light coming into this photosystem 1. It's going to do the same thing. It's going to come in. It's going to accept. It's going to go to these pigment molecules, and it's going to bounce around until it hits the P700. So as this light comes in, it excites these molecules moving into this P700. The P700 then is going to give an electron to the primary electron acceptor. As it gives that electron away, is it reduced or oxidized? It's oxidized, and then it's going to be reduced by these electrons coming down the electron transport chain. Okay, so when P680 gets oxidized, it accepts electrons from water being split. When P700 becomes oxidized, and becomes P700 plus, it can then accept the electrons from the electron transport chain. So it's going to accept Oops, that should be my. So it becomes oxidized and then it becomes reduced by those electrons moving down the electron transport chain. So P700 is the electron acceptor instead of oxygen. When it donates the electrons, it's going to push the electrons up to that primary electron acceptor. And we have another electron transport chain. So there's two electron transport chains in this case. So this second electron transport chain is going to give these electrons to another mobile electron carrier, which transfers it to an enzyme, NAD plus reductase. ACE tells us it's what? Enzyme reductase tells us it's going to do what? Reduce. What is it going to reduce? NADP plus, because this is a little bit different than the other one. It's going to reduce NADP plus, and it's going to form NADPH, which is a full electron carrier. So this electron change is different in that there's no hydrogen ions being pumped. So we're not moving hydrogen ions down this chain. We're not producing any ATP. It's literally just moving electrons. It's transferring electrons to this NADP plus reductase to make NADPH a full electron carrier. So step 8 is to fill the electron carrier NADP plus. The benefit of putting the electrons into NADP plus to make NADPH is these electrons have more energy than the original electrons that were in water. So we've taken electrons from water, we've moved them through this whole complex photosystem 1, passed them down an electron transport chain, transfer them here, bounce them up again, and transfer them down to here. And you can see they're at a higher energy level than they were before. So now we have high energy electrons available in a full electron carrier, and we have ATP. So your book does not go into the numbers, but we're just going to pretend it always comes out even. So in the light reactions, what are we making? We're making ATP. So this is what we're getting out of it. And we're just going to pretend that we make the exact number that we need for the next thing. So there's 9 of them. I'm going to be more efficient. So we're getting ATP molecules. What else are we making? What are we making? NADPH. Yes. So we're filling electron carriers. 1, 2, 3, 4, 5, 6, 7, 8. Oh, I don't have to take 9. We're pretending we're making the exact number that we need, which is 9. This does not work out as nicely as the restoration in terms of space on the wall. Okay, so on your yellow chart, fill in what we're making. We're making ATP, and we're making full electron carrier. So in addition, we also made oxygen, but the exciting part of this is not, well, I mean, oxygen is exciting in that we need to breathe it, but what's going to carry on to the next thing is we're making NADPH and ATP. What? Yes, these are the light reactions. So in the light reactions, we're going to move through photosystem 2 and photosystem 1. We're going to make ATP and we're going to make NADPH. Have we made any glucose yet? Any carbohydrates? anything with carbon. We haven't even touched carbon dioxide yet. So the only thing that we've dealt with in the reactants is the water. We split the water to get the electrons, and we use those electrons to make ATP and NADPH. So this shows it in a slightly different way. Okay, I just love it. Someday I'm gonna master technology. If it doesn't master me first, I just log myself out. That's good. Okay, there we go. But it's not, there's working.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: photosystem 2, cytochrome B6F, photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem 2 complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem 2, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem 2 are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem 1 complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP, NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP-NADP After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
I'm just waiting to see if it's gonna start a new video. Okay, so I like the way that shows it. They use a couple different terms. The resonance energy is just the vibration of the pigment molecules that transfers the energy, and they use the term lumen instead of intermembrane space. But again, still the idea of creating the gradient. So this is just showing you again an overview. We have a photon coming in, we take this electron, we power it up. It's going to move down the electron transport chain using some of its energy to make ATP, and then another photon is going to excite it again. And we end up with these electrons in NADPH that have more energy. This is also showing you just the process of the ATP synthase as we move through the chemiosmosis process. This was step 5. And again, same idea as what we talked about. We've got the mobile electron carriers that are moving the electrons through. Oh, sorry. Someday. Mhm. Why does it go back to that? So you didn't even see this picture. There you go. So this is showing you, this is just a mechanical drawing, showing you how energy is added to move through to get more oxygen, more energy in the electrons. Okay, so this is chemiosmosis in the chloroplast, which looks very similar to the way it does in the mitochondria. We have the movement of electrons and the pumping of hydrogen ions. Wait, I said that, sorry, the lumen is the thylakoid space versus the stroma, which is the space around the thylakoids. So we're going to pump these hydrogen ions in. We're going to get a high hydrogen ion concentration here. Those hydrogen ions then can move through the ATP synthase. And that's going to provide the energy needed to make this bond between ADP and PI to get ATP. And this is just showing the continuation of the system. So this is PS2 over here. It's not in here. Continuation of the light reactions with the electrons and what's happening to the electrons to the point that we're getting this NADPH over here with the higher energy electrons. With this high hydrogen ion concentration, it actually changes the pH in there to the point of it can lower the pH by 3. So it makes it more acidic. So if you start at a pH of 7, it could reduce the pH down to 4. So we get this, it's a hydrogen ion concentration gradient, but also a pH gradient really of a low pH and a higher pH. And as those hydrogen ions move, that will more equal out the pH again. So this is still phosphorylation when we're doing this, but in this case, because we're not using in the same way, it's similar to the oxidative phosphorylation, but we call all of this photophosphorylation because it's light that's providing the energy ultimately for the phosphorylation. There's no organic molecules involved in this in any way, so that makes it different from the electron transport chain and cellular respiration. We have no organic molecules, no carbons, no nothing. But similar ideas in how the chemiosmosis works in the electron transport chain. So you can think about the similarities and the differences between those. All of these, our two main products, the ATP and the NADPH, are going to end up going on to the next set of reactions, which is the Calvin cycle, which we're gonna start, but I don't anticipate getting through. And that's okay. So light reactions, we take water, we make oxygen, we make ATP, and we make NADPH. So what is the reactant that's going to be involved here? CO2. So this is our CO2. It's going to come into the carbon cycle. And what is going to be our product? It's going to be a carbohydrates. And the specific carbohydrate that we're really making is called G3P, which is glyceraldehyde 3-phosphate. This has 3 phases, so we might talk about the first phase. Maybe we might get further than I think. That's the overview. Let's go to the details. So this is the details of our Calvin cycle. So it does not show up on me. So at the top, you can see this molecule. There's something on my screen that is not on your screen. There we go. This is carbon dioxide. How many carbons are in carbon dioxide? 1. So we're going to start thinking about our carbons too. We have a 1 carbon molecule coming in. Carbon dioxide is our 1 carbon molecule. We are going to combine it with another molecule, which is a 5 carbon molecule called Rubisco. So this is a 1 carbon, and we're combining with a 5 carbon molecule. So now we have a what carbon molecule? 5 plus 1 is 6. Good. So we've got a 6 carbon molecule. This phase where we're combining the CO2 plus the Rubisco is called carbon fixation. So we're taking carbon that's in CO2 gaseous form, and we're making it into a 6 carbon solid carbohydrate form. So we're fixing it. You've probably maybe heard of nitrogen fixation. There are bacteria that can take nitrogen out of the air and make it into a usable form. This is the same idea. We're taking carbon out of the air and making it into a useful organic carbohydrate molecule. This is going to happen with the Calvin cycle 3 times. So remember when we talked about the citric acid cycle? We went through the citric acid cycle twice because we had 2 acetyl-CoAs from our glucose that we split. This is going to happen 3 times. So we really have 3 carbons that we're going to deal with because we're going to go through the Calvin cycle 3 times. So we're going to keep track of our 3 carbons because we can't lose matter. But each of those we're combining with a 5 carbon molecule. So you'll see a lot of numbers on this diagram. We're going to make 3 of these molecules. This is a 6 carbon molecule that's pretty unstable. So it's actually going to split. So if it's a 6 carbon molecule and we split it, how many carbons are going to be in our next molecule? 3. So we're going to have 2 3 carbon molecules. Again, I'm not concerned with you memorizing the names of all these molecules. We're not even talking about the names of all the molecules, but understanding the basic idea of we're taking a 1 carbon, we're putting it with a 5 carbon, we're splitting it into a 3 carbon. So we split into this 3 carbon molecule. Did I talk? Rubisco, sorry, I didn't tell you this. It's called, we abbreviate it as RUBP. Sorry, no, that's the enzyme that combines them. So the Rubisco enzyme, RUBP, is possibly the most abundant protein on Earth. Because this is happening in all the plants and all the cells of all the plants. Remember, each mesophyll has 30 to 40 chloroplasts in it. So this is happening in all the cells and all the plants, in algae and cyanobacteria and all those. So this is possibly the most abundant protein molecule on Earth. So we're going to take this process. You'll notice this molecule has a phosphate on it. We can then use energy to take off that we're going to use energy from ATP. We're going to break off a phosphate and we're going to add another phosphate. So we have 1 phosphate. We're going to get a 1 3 biphosphoglycerate molecules. So now we have 2, yeah. Rubisco is the name of the 5-carbon molecule. I did not say that clearly. So the 5-carbon molecule is Rubisco. RUBP is the enzyme. Rubisco biphosphatase is the name, but we abbreviate RUBP. So that's the enzyme. The enzyme is what adds the carbon to the Rubisco. And then, so then we have our 6 carbon. So then another enzyme will split it, but we're not going to talk about the names of all of them. RUBP is just the really important one that we're going to talk about. So we're going to take the phosphate from the ATP and add it to this 3-carbon molecule. So now we have a 2-phosphate glycerate, still a 3-carbon molecule. You'll notice they're using the number 6 here because we're going through this 3 times. So this molecule is going to split into 2, but there's 3 of them coming in, so they use the number 6. Okay, so that's carbon fixation. The next one we'll just talk about, we'll finish there. We'll talk about phase 2 and phase 3 on Monday. What do you need to do when you leave class today? Take the quiz or write down when you are going to take the quiz. Okay? Don't forget to sign the attendance sheet if you didn't, and to give me your homework if you did not.